Hello students, this is our chapter Molecular Basis of Inheritance and today we'll study of Human Genome Project. The Human Genome Project, commonly called HGP, each individual has an identity that is due to one's genetic makeup. No two individuals are similar except monozygote twins because they differ in their genetic makeup. Difference in genetic makeup are due to differences in nucleotide sequences of their DNAs. It was therefore always an ambition of scientists to map human genome. Advances in genetic engineering techniques made it possible to isolate and clone DNA pieces and determine nucleotide sequences of these fragments. Therefore, in 1990, US Department of Energy and National Institute of Health embarked and coordinated on a project of sequencing human genome called HGP or Human Genome Project. Wellcome Trust, United Kingdom, joined the project as a major partner. Later on, Japan, France, Germany, China, and some other countries also joined it. John Craig Venter was the first scientist to sequence human genome. It was launched on 1st October 1990. The project was completed in 2003. Thus, it was a 13-year project. So, Human Genome Project has been called a mega project because of a huge cost estimated to be 9 billion US dollars and very large number of base pairs, that is 3 into 10 raised to power 9 base pairs, to be identified and sequenced. A large number of scientists, technicians and supporting staff are needed. Storage of data generated which requires more than 3,300 books, each with 1,000 pages and each page having 1,000 typed letters. However, high-speed computerized devices for storage, retrieval and analysis of data made it easier to do the same. So the science of bioinformatics also developed during the period and helped Human Genome Project. So what is bioinformatics? Bioinformatics is a new field in which computer hardware and software technologies are developed and used to gather, store, analyze and disseminate biological data, images and other information. So these are the goals or aims of the project. So identification of all the approximately 20,000 to 25,000 genes in human DNA, first it was. Second is to determine the sequences of 3 billion chemical base pairs that make up human DNA. Then third one is to store this information in databases. And fourth is to improve tools for data analysis. And fifth is transfer related technologies to other sectors such as industries, etc. And then to solve any ethical, legal, social issues, that is called LCELSI, that may arise from the project. Then bioinformatics, close association of human genome project with the rapid development of new area in biology. There are two approaches for sequencing human genome. First is expressed sequence tags, that is ESTs. Identify all the genes that are expressed as RNAs and sequence the same. And second one is sequence annotation. The whole genome, including both coding and non-coding regions, is first sequenced. Later functions are assigned to different regions. In Human Genome Project, sequence annotation has been carried out which involves following steps. So the whole DNA of cell is isolated and broken randomly into fragments. They are inserted into specialized vectors like BAC, bacterial artificial chromosomes, and YAC, that is yeast artificial chromosomes. The fragments are cloned in suitable hosts like bacteria and yeast. So polymerase chain reaction is that by which we can amplify the number of fragments of DNA. Can also be used for cloning or making copies of DNA fragments. The fragments are sequenced as annotated DNA sequences and the sequences are then arranged on the basis of some overlapping regions. It necessitates the generation of overlapping fragments for sequencing. computer-based programs are used to align the sequences. The sequence were then annotated and assigned to different chromosomes. All the human chromosomes have been sequenced, 22 autosomes and X and Y chromosome. Chromosome number 1 was last to be sequenced in May 2006. With the help of polymorphism in microsatellites and restriction endonucleases recognition sites, the genetic and physical maps of genome have to be prepared. This is chromosome number 1. It has maximum number of genes, that is 2968 genes. This is Y chromosome, which has minimum number of genes, that is 231. So there are maximum and minimum genes for the human chromosomes. And next topic is our salient features of human genome, which we'll be studying in the next lecture.